Hello everyone, this is Shinuja, Assistant Professor, Department of Biotechnology, St. Josephs College of Arts and Science for Women, Vasur. Today topic is Tissue Engineering of Skin. Tissue engineering is a scientific field concerned with the development of biological substitutes capable of replacing diseased or damaged tissue in humans. The term tissue engineering was introduced in the late 1980s. By the early 1980s, the concept of applying engineering to the repair of biological tissue resulted in the rapid growth of tissue engineering as an interdisciplinary field with the potential to revolutionize important areas of medicine. Tissue engineering integrates biological components such as cell and growth factor with engineering principles and synthetic materials. Examples of tissues that are candidates for tissue engineering include skin, cartilage, heart, and bone. The production of skin substitutes has played an important role in improving the success of skin graft surgeries, especially for complex wounds such as burns. Substitute tissues of the renal system, including urinary bladders and urethras, have also been engineered and transplanted successfully, thereby broadening therapeutic opportunities for complicated renal disorder. Three key components of the tissue engineering process. Key component of a tissue engineering process are a tissue or cell sample that constitutes the seed and starting point of the process, and secondly a scaffold that holds and supports the seed and steers its growth into desired geometrical shape and location. A third important component is an environment of signal substance and nutrition, etc., that steers the growth cell culture or tissue seed towards the desired biological properties, including both geometrical size, shape, and biofunctional properties. A bioreactor steers the shape and functional properties of the tissue. The tissue engineering process is often implemented in a bioreactor designed to steer both the geometrical shape and the biofunctional properties. In addition, the bioreactor also provides pressure and temperature control and specific motion program like agitation and rotation. The process of tissue engineering typically involves several steps. First one is cell sourcing. Cells are harvested from the patient's own body, that is autologous from donors, allogenic, or may be derived from stem cells. Biomaterial selection. Biomaterial serves a scaffold to support cell growth and provide the necessary mechanical and biochemical clues for tissue development. These materials should be biocompatible, biodegradable, and possess appropriate mechanical properties. Cell seeding. Cells are seeded onto the biomaterial scaffold either in vitro, that is outside the body, or in vivo, that is inside the body. The scaffold acts as a framework for the cells to attach, proliferate, and differentiate. Cultivation and differentiation. Cells are cultured under controlled conditions, which may include the provision of specific growth factor, mechanical stimulation, and or biochemical clues to guide their differentiation into the desired cell type. Maturation. The engineered tissue is allowed to mature and develop functional properties similar to native tissue through continued culture and optimization of environmental conditions. Implantation. Once the tissue has reached an appropriate level of maturity, it can be implanted into the patient either alone or in combination with existing tissues to replace or repair damaged or diseased tissues. Tissue engineering of skin. Tissue engineering of skin involves the development of artificial skin substitutes that can be used for wound healing, skin graft, and various dermatological conditions. The ultimate goal is to create a functional equivalent to native skin that can promote regeneration and repair of damaged or diseased tissue. Tissue engineered skin typically consists of three main components. The first one is epidermal layer. This layer is responsible for providing a barrier against environmental factors and pathogen. In tissue engineered skin, the epidermal layer is usually composed of keratinocytes, the primary cell type found in the epidermis. These cells are seeded onto a biocompatible sac and cultured to form a stratified epithelium resembling the native epidermis. Next one is dermal layer. The dermis provides structural support, elastic